So today I'll be presenting our project, High Cervical Epithelial Expression of Interferon-epsilon in HIV-1 exposed seronegative sex workers. Now, as a brief primer on highly exposed seronegative individuals for people who are not initiated, these can be broadly split into two different groups. These are individuals who remain uninfected after blood-to-blood contact, or individuals who remain uninfected after persistent sexual exposure. Good example would be looking at individuals in serodiscordant relationships with an HIV-infected partner, or, as we'll be talking today, sex workers who remain seronegative despite drawing clientele from an area of high HIV-1 prevalence. So, to date, there have been many different correlative studies looking at factors that might give ESNS their protection or their resistance to infection. But to date, there's been no true found correlative protection. But one study that caught our interest 13 years ago from the Frank Plummer group was one that actually looked at individuals who seroconverted after attaining this protected status. And of the factors that they looked at that associated with this loss of protection, it was either a break from sex work of 2 months or longer, or a reduction in the number of clients before their infection. So from this, we hypothesized that perhaps a persistent exposure, vaginal exposure in these sex workers would generate an ISG response within their vaginal mucosal tissue, which could provide a barrier to infection. So to pursue this hypothesis, we got a cohort of sex workers and control women from the San Juan, Puerto Rico metropolitan statistical area, which is an area of high HIV-1 prevalence within the United States. The control group was from a family planning clinic at the University of Puerto Rico, and the sex workers were from the general area. The sex workers on average had 8.5 years of sex work and averaged about 8 unprotected coital acts per week. This, of course, they had additional acts that were protected. To assess this group, we looked at HIV-1 memory responses, including CD8 T-cell memory responses to HIV-1 gag, and we also looked at cervical vaginal IgA and IgG specific to HIV-1. As far as systemic indicators, we looked at PBMC subsets, specifically looking for activation levels, and looked at serum cytokine levels by Luminex. And to address our hypothesis, we looked at ectocervical biopsies doing gene arrays and tissue staining. And here, just a beautiful picture from San Juan, Puerto Rico. It's a really beautiful place if you ever have an opportunity to visit. So for a little more detail as to the population of interest, there was very little oral contraceptive use amongst the sex workers. The sex workers tended to use some form of sterilization to prevent pregnancy. Not surprisingly, there was a higher level of STDs amongst the sex worker group. And importantly, we also looked at estradiol and progesterone within the group and found very little difference between the sex workers and control individuals. So looking at those systemic indicators, one of the strongest findings was lower T-cell activation, both in the CD4 and CD8 T-cell compartments. And here we found much lower CD38. We did look at DR, but we didn't see any significant differences there. In addition, we also found that 5% of the sex workers had some detectable level of either mucosal antibody responses or HIV-specific T-cell responses. Both of these findings seem to be in line with previous studies looking at HIV-1 exposed seronegative individuals. Next, we wanted to look at the tissue itself, and this is something that has not been done very frequently within HESN cohorts, just taking the tissue and staining it for cellular infiltrates. And in this case, we looked at CD123 for PDCs, CD68 for macrophages, and CD4 for CD4 T cells within the ectocervical biopsies. And as you can see here, each of these were elevated within the sex worker group, with the highest being CD4 T cells. Next were macrophages, and finally were the CD123 PDC. Of interest, the CD68 macrophages and CD4 T cells seem to locate more closely to the epithelial layer, while the PDC seem to be more distributed throughout the lamina propria. Next, we took a subset of these individuals to do our gene array, and as you can see, looking at the principal component analysis 1 and 2, which accounted for about 43% of the variability among significant genes, the sex workers clearly segregated from our control group. So from there, we decided to do an Ingenuity pathway analysis on these significant genes and look to see what biological functions were impacted within this group. Here we're showing 1, 4, and 5 of the significant pathways that were changed. And notably, we saw functions related to homing, chemotaxis, and leukocytes being reduced in sex workers. And we also saw alterations in cell-mediated immune response pathways. Next, we decided to look at all the significant genes, and we pulled out immune-associated genes within our gene array. And as you can see, I don't put the details, but you can see, but the sex workers had lower level expression of multiple different immune-related genes associated with ISGs, activation, and migration of immune cells. But of interest, there was a small subset of genes that were upregulated within the sex workers. And the most notable of those was interferon-epsilon. Now this caught our attention because interferon-epsilon has been shown in papers published last year to be involved with the protection of the female reproductive tract from bacterial and viral infections, at least in the mouse model. So we decided to pursue that further and do actual staining to look for the protein within the tissue. And here we see 2 examples, one from our control group and one from our ESNS sex workers. And very interestingly, you can see that the staining seems to be confined to the basal and parabasal layers of the tissue. When we quantified this, we saw importantly, that we had a significant level of interferon-epsilon in the sex workers as compared to the control group. And this strongly correlated with our array data, really convincing us that the interferon-epsilon was higher within the sex worker group. But then, of course, we had the question, well, what was causing this higher level of interferon-epsilon? Research done last year suggested that interferon-epsilon was under the control of estradiol and other female hormones. However, as I showed before, there was no significant difference in that between the 2 groups. Closer inspection of those papers actually highlighted that was a potential role of semen and the control of interferon-epsilon within the tissue. So to investigate that, we combined both the control group and our sex worker group and split them apart based on sex acts. So here you see sex workers or control members who had 1 or more sex acts in the week prior to sampling, and those who had no unprotected sex acts in that prior week. As you can see, a much tighter separation between the groups, with interferon-epsilon within those who had sex recently being almost 3 times higher than those who did not. To further pursue that angle, we looked at ECT1, which is an ectocervical cell line developed in the 1990s, and exposed that to a 5% seminal plasma solution. And as you can see here, looking at immunofluorescence, it increased or enhanced the level of interferon-epsilon expression within the cells. We also confirmed this using RT-PCR, which also showed a 2 to 3-fold higher level of expression. So for our conclusions, what we have is a low level of HIV-1 specific memory within PBMC, reduced T-cell activation, which did not correlate with serum cytokines, which I didn't show, or hormone-based contraception. Within our tissue, contrary to our hypothesis, we found no evidence of an ISG signature within the mucosal tissue, but there was a downregulation of multiple families of cervical-associated immune genes. We also found high epithelial expression of the type 1 interferon-epsilon, and our in vitro data seems to support a role of semen in the regulation of this interferon. So as a model, what we think is happening is with seminal plasma or seminal fluid come into the compartment, which would induce both a recruitment of macrophages and CD4 T cells and other immune cells, but also induce the expression of interferon-epsilon, which theoretically could be involved in infection resistance through type 1 interferon mechanisms. And if this is the case, then it's possible that interferon-epsilon joins a growing list of known HIV antimicrobials. So firstly, I would like to acknowledge all of the female participants, both the sex workers and our control group women who participated in this study. I would like to thank my mentor, Luis Montaner, and all the individuals in our lab who have helped in this project. And of course, our collaborators at the University of Puerto Rico, the University of Nebraska, and Duke University. Thank you for your attention.